Dear students, here is another lecture about classics in total synthesis, in which we will study about estrone. Estrone is basically a sex hormone, which is also a class of steroids. So, before going into the details of estrone, we will briefly discuss here what are the steroids. So, basically, steroids are the organic compounds as shown here in the figure, which contain this type of skeleton in which three six-membered rings A, B, C, and a five-membered ring D is attached together. This type of ring containing compounds mostly known as steroids, and other functional group attached to this skeleton produce different kind of steroids. So hundreds different kind of steroids are present in plants and animals and fungi, which have their different roles. So basically all steroids in animals and plants are synthesized from common precursor saccharine, which is a triterpene derivative, which we will discuss in the next few slides. So basically, all steroids contain this type of core structure, as you will see on this slide. So steroids are basically biologically active compounds with four ring arranged in a specific molecular configuration, and they perform basically two important functions. First one is they are important component of cell membranes, which alter membrane fluidity and as a signaling molecules. So now move to the next slide. This is the saccharine, which is a precursor of many steroids. uh, this saccharine is converted into lanosterol, which further converted into cholesterol through biosynthesis. And this cholesterol is further a precursor of a different type of steroids such as gonane, which further produce sterol. So this is simple illustrated diagram from which you can uh, understand how these steroids are produced. So what are the basically steroids? Basically steroids are the hormones, uh, and these hormones further classified into two different classes. The first one, which is known as corticosteroids, these are typically produced in the adrenal cortex, while the sex steroids are produced in the gonads or placenta. Sex hormones are also known as sex steroids, gonadocorticoids, or gonadal steroids. And these sex hormones are further classified into three different classes. First one is androgens. Mostly, they are present in higher quantity in male, uh, while the estrogens are present in the female, while progestogens are further regulatory enzymes present in the both. So here is the brief history of the estrogen, how estrogen word evolved. Basically, it evolved from the oestrus and genus. Oestrus mean a periodic state of sexual activity in female mammals, while genus is generating. So basically, the estrogen is the word is derived from these two words. So it is also referred as oestrine, and somewhere it is also still used as estrogen. So what are the estrogen or oestrogen? Basically both words represent the same class of steroids. They are basically sex hormones are responsible for the development and regulation of female reproductivity system and secondary sex characteristics. Further, estrogens are uh, classified into four further steroids. First one is the estrone, second one estradiol, third estriol, and fourth one the estrotetrol, which is only produced during the pregnancy. So in 1929, first the estrone was isolated, uh, uh, the first estrogen discovered by Adolf and Edward Dozy independently. Shortly following their discovery, other estrogens, both natural and synthetic, were introduced for the medical use. So they also have a significance in the medical field. For example, estriol glucuronide, which is basically estriol, is combined with glucose. Similarly, estradiol benzoate conjugated with benzoate conjugated estrogens, premarin, diethyl bestrol. All these have medical applications, different medical applications, which you can search out in the literature. So estrogens are synthesized in all vertebrates and even some insects. So their presence in both vertebrates and insects suggests their estrogenic sex hormones have ancient evolutionary history. So qualitative estrogens circulate at lower levels than androgens in both men and women, while estrogen levels are significantly lower in males than in females. So estrogens are present in higher quantity in females. Estrogen nevertheless has important physiological roles in male. Uh, like all steroid hormones, estrogens readily diffuse across the cell membrane. Once inside the cell, they can bind and activate other estrogen receptors and turn modulate the expression of many other genes and functions. So here was the briefly discussion about estrogens and their brief history. So let's move to the next slide. Here are the some few, because here we will discuss about the estrone retroanalysis and its total synthesis. So first of all, we will discuss here a simple some important reactions which leads toward us to the leads us toward the synthesis of estrone, and the and a brief you can say that these reactions help us to construct different uh, steroid structures. So first of all, we will discuss here the intermolecular Diels-Alder reaction. So as you see here, we can construct a cyclohexene of our desired molecule. Further, we can also intramolecular Diels-Alder reaction in which we can also construct a two-ring system within the molecule. And in the next slide, here is another important cyclization reaction, prototypical 2 plus 2 plus 2 cycloadditions, where we can cyclize alkenes, alkenes and alkynes. So here you can see ethene derivatives can be cyclized to form cyclohexene. Similarly, ethine and ethene derivatives can also cyclize oligomerized to form cyclohexene derivative. And similarly, ethylene derivatives can also lead toward the synthesis of benzene type products. So these are the simple reactions. So here, whenever you want to construct a cyclic ring member, so you can use Diels-Alder reaction, cycloaddition type of reaction strategy to synthesize different type of cyclohexanes. So here is another exemplary reaction which is used for the construction of benzocyclobutene and subsequent reaction. So all these type of reactions are used frequently used in the steroid skeleton synthesis. Basically, these are all these reactions help us to construct different type of ring structures, which we will use all these strategies in the synthesis of estrone. So here is 1,5-hexadiene, which is oligomerized with, or you can say cyclized with bis trimethyl silyl acetylene, which is represented by BTMSA, and cyclopentanyl cobalt carbonyl dicarbonyl is used as a catalyst. As a result, we get this type of benzene cyclobutene type product with 60% yield, and further elimination of these methyl silyl ethers leads to the product 12. And further, this product on heating at 200 degrees centigrade, this cyclobutene ring is cleaved and converted into orthoconidomethane, which is further act as a uh, diene in a Diels-Alder reaction. And here is our dienophile. As a result of this Diels-Alder reaction, we get this type of cyclic ring chain. So here you can see that the cyclization of acetylene derivatives leads us to the synthesis of this bicyclic ring member, which contain benzene ring and cyclobutene, which further we converted into this type of diene, and further reacted with this dienophile, which is an which further convert us leads to the product number 11. And here is another important significant reaction, which is also frequently used for ring construction of steroids. So observes synthesis of kilodonine. So here you can see that this type of ring structure is can be synthesized easily by oligomerization of acetylene derivatives, and further by coupling this type of molecule, we get this product, which further on uh, treatment with orthoxylene at 120 degrees centigrade and intermolecular Diels-Alder reaction, intramolecular Diels-Alder reaction take place, and we get this type of product, and which is further through different steps is converted into our target molecule kilodonine. So here are different other ring construction, different strategies from which you can synthesize a different type of ring structures. So if you connect the acetylene derivative with this type of chain ether linkage, you will get after treatment with cyclopentadiol cobalt dicarbonyl in the presence of bis trimethyl silyl acetylene and reflux, you will get this type of product. Similarly, with slight different variations, you will get different type of products. So basically, using these different strategies, as I earlier discussed, you can construct different ring structure models, which will help you to synthesize different kind of steroids. So now come to the retroanalysis of Estrone. So here is the Estrone structure. So first of all, we will do the cleavage at this with position represented with dotted line. So this is basically intramolecular Diels-Alder reaction. As a result, you will get this type of product or intermediate, which is represented by number 14. And this intermediate exists into exo transition state A or exo transition state B. Basically, in exo position, both uh, diene and dienophile uh, substituents are in the opposite plane, while if they are in the same plane, you can say they are in the endo position. So after the cyclization, or you can say cyclooligomerization, you will get this bis trimethyl acetylene and this type of derivatives of after the alkylation or cleavage at this position, which further you can cleave this Michael addition reaction, and you will get the methyl cyclopentanone, and this you can say acetylene and derivatives, and further this alkylation part. And this alkylation part we can get from uh, epoxide type of compound. Here is ethylene oxide and 1,5-hexadiene. Now come to the synthesis of estrone. So here we can use 2-methyl cyclopentanone, which is further reacted with Grignard reagent in the presence of cuprous iodide. And this cuprous iodide will change the addition of Grignard reagent. Now, mostly cases, Grignard reagent add at this position, as I should represent here. Wait a minute, just. So if you did not use this cuprous iodide, then this Grignard reagent will be added at this position. However, this the presence of this salt alter its addition. As a result, it will add in 1,4 Michael addition type. Reaction will take place, and as a result, this methylsilyl chloride also protect this keto functional group. As a result, this alkylation will take place at this position. Otherwise, if you did not use this, then this Grignard reagent will add at this position. As a result, you will get this type of product. So in the presence of cuprous iodide, you will get this product. Further on treatment with base, you will remove uh, trimethyl silyl ether and convert it into this type of intermediate, which further alkylated with this group. And this 17 is synthesized by 1,5-hexadiene, which is coupled with ethylene dioxide. As a result, you get this type of product, which further on treatment with tosyl chloride in the presence of pyridine and sodium iodide and in acetone, you will get this type of product. Further on coupling this reaction with this product, you will get this type of product. And further on treatment with is trimethyl silyl acetylene, this trimethyl silyl acetylene, you will get this type of cyclized product, this, which further on treatment in decane reflux, you will get the next. We will move on to the next slide. we will get this 14 um, type of molecule, and which further through intramolecular deal that reaction is converted into 13, and which further treated on with tetrafluoroacetic acid in carbon tetrachloride at minus 30 degrees centigrade and lead tetrafluoroacetate, we will get our estrone. So here was the total synthesis and retrosynthetic analysis of estrone. Further, you can, if you have any type of question, you can text on the WhatsApp, or you can also email me. Thank you.